So Dr. Scott Williams received his PhD.
Dear doctor, your time is up.
Not Scott, of course. Received his PhD from Washington University in St. Louis. He was awarded a 2012 American Association for the Advancement of Science Fellow. He's worked as a section editor for PLOS Genetics. Is currently a section editor for Genomics. Dr. Scott is a member of the Executive Committee of the African Society of Human Genetics from 2013 to date, and also part of the program committee for the Pacific Symposium on Biocomputing. So, welcome, Dr. Scott Williams.
All right, thank you. And I would like to thank the other members of the organizing committee for allowing me to speak, because I'd like to thank the whole organization for having me back. The other thing is I'd like to thank all of you for sticking around. It's been a long, but very actually fruitful meeting, I think. And I think there's been a lot of really great science presented. So what I want to talk about is kind of going back with a slight twist to what Charles Rotimi opened up on a couple days ago about the role of diversity and why study African populations. Now, I'm not going to talk about African populations per se, but I am going to talk about the impact of the diversity that they generate globally and how that affects it. But I'm going to change it a little bit. I'm going to ask the question, how does admixture between human populations impact disease risk or susceptibility? And then the second story is, how does admixture in the bacteria or potential pathogens impact the pathogenicity of these bugs? And so I'm going to talk about Helicobacter and human coevolution. Helicobacter pylori is a curved rod-shaped gram-negative bacterium, and if you look at the picture on the lower right-hand corner, you see those little black squiggles. Those are the Helicobacter in the human mucosa. Now, for those who know, Barry Marshall and Robin Warner won the Nobel Prize for discovering, in fact, that Helicobacter causes gastric ulcers. It also causes gastric cancer. And gastric cancer is the primary cause, and I mean cause, not association. It's usually necessary but not sufficient. About 10% of cancer mortality is gastric cancer, and it's the second leading cause of cancer worldwide. Now that said, half of the people on the planet are infected with Helicobacter pylori, but most of us, or those of us who are positive, will not get it. And in the low and middle income countries, that infection rate is much higher. Yet of those who are infected, about only 1% get cancer. So the question really becomes, if you have a commensal pathogen or bacterial infection, what makes it pathogenic? Under what conditions does that actually become a pathogenic infection? As in most cases here, it isn't. Same thing's true of Mycobacterium tuberculosis. About a third of the world's population has been exposed, and there are only 9 million cases a year. So what happens when you get infected? If you have a normal mucosa, everything goes along fine. You get a H. pylori infection, you can go down a couple of pathways. Usually if you have, and those little ovals, CAG-A- and BAC-A, are actually bacterial genes. If you usually get a non-atrophic gastritis with a CAG-A-, but if you have a CAG-A positive, you have the potential to go down from either through non-atrophic gastritis all the way down this cascade to cancer or to an ulcer. And what we're going to talk about today is actually the movement down this cascade. So the other thing to remember and to note is that Helicobacter pylori has infected humans since before they left Africa. And in fact, it migrated out of Africa in the guts of humans. And that's very important because, in fact, as we've discussed about all this geographic differentiation in the human population, there is substantial geographic variability and defined countenance in the Helicobacter genome as well. So here's from a paper of Falouche et al. In Science in 2003, looking at the putative modern and ancient migrations of Helicobacter. It turns out they really mimic the movements of humans very well because that's where they exist. So the other thing about Helicobacter is it's predominantly transmitted from mother to offspring. It's inherited akin to mitochondrial DNA. And because of its kind of consistent co-infection, it's effectively a prudent parasite. It sticks in the guts of most or half of all humans, 90% or so in the developing world, and in fact, it doesn't cause disease most of the time. So what affects this virulence is the ability to actually cause active disease. One of the things that's very important in terms of the evolutionary history of it is, in fact, the vertical versus the horizontal transfer. Because you inherit it, you're inheriting with the genome of your mother. So one could postulate that there's been actually the opportunity for coevolution to occur such that the genome of the bug and the genome of the people have actually come to a reasonable benign coexistence. So it should be able to co-evolve to being relatively benign. It is relatively benign. Can we ask the question, is what's driving that state? So to reinforce it, about 75% of the people who are infected with H. pylori have absolutely no clinical presentation at all. But nearly all adults in the developing world are infected. In fact, the Egyptians in this room, approximately 90% have an H. pylori positive. Now, one could say, if it has the potential to cause cancer, why don't we just eradicate it from everybody? Well, there are a couple of reasons why not. One is, when you eradicate it, you actually increase the probability of having gastroesophageal reflux disease. You probably don't want that. There's also a theory which has some substantiation, is that in youth, it prevents from asthma. So when you're young, it lowers the risk of asthma. When you're older, you can get gastritis all the way down through gastric cancer. So we would like to know who we want to eradicate from, who are the people most at risk. So we know a little bit about the genetic factors in the bug that cause increased risk. CAG, as I mentioned, is one of them. The other is VAC-A toxin. But neither is necessary nor sufficient for gastric cancer. They increase the risk statistically significantly and probably clinically relevant changes, but not huge changes. So, as we ask these questions about the determinants of virulence, we actually have to go down a series of looking at what is the natural history of this globally. And the interesting thing is that the rate of infection is not predictive of the rate of disease, of the prevalence of disease. And that's somewhat opposite of what you'd expect with an active infection. You know, there is evidence for host and pathogenic genetic variation, as well as environmental things such as diet. But a few years ago, these terms were coined, and this is where the African part comes in. So the variance in virulence by geography is not a strong correlation. This gave rise to the African and the Asian enigma, and more recently, the Latin American high altitude enigma. What that means is these three locations have high rates of infection, the first two at least, and many of the African and Asian populations have low rates of disease. And Lunet and Barros kind of coined the term about a little over 10 years ago and said some countries from Africa and Asia present low gastric cancer rates and high H. pylori prevalence, which has been labeled the African and Asian enigmas. Now, to give you an idea, this is some data from their paper. So you look at, for example, Egypt. 88%, the prevalence of infection is 88%. But the gastric cancer is only 3 per 100,000, whereas Italy, 45%, but 15 per 100,000. And Germany, even lower and are comparably high. This kind of lays out the enigma. So what's causing this enigma? And let me give you a little bit of history about H. pylori. I mentioned already that there is this kind of geographical provenance, and people usually determine the geographic origin of an H. pylori strain by sequencing 7 housekeeping genes. It's called multi-locus sequencing type. And this is we've seen a lot of structure plots. This is a structure plot without admixture, giving the Sahel, East Asia, Asia 2, Europe, Northeast Africa, and Africa 1 and Africa 2 variants or strains. This is a model that doesn't allow us to look at the multiple ancestries of the strains. But in fact, there are distinct evolutionary histories that correspond to human movement. Now, the other thing about, not only do you have distinct geographic continents, but it's a really bizarre genome. 6% of the genes are just absent in some strains. They're not universally the same. The other thing is really a recombinant bug. If you get infected with two strains, you will ultimately end up with a chimeric strain, an admixed human populations. And there are lots of inversions and translocations. It actually turns out to be a real bear to align the sequence of this relatively small genome, because things don't always have the same shape, the same topography. So we can look at admixture in the isolates, and we can, but that's usually ignored in typing. So here, if we take the same thing, the top panel is what I showed you earlier. The bottom panel is if you allow you to examine admixture of those same isolated strains. And you can see that the HP Europe strain is predominantly green or European, as is the North African. The HP Africa 1 is predominantly blue, whereas in the analysis where you don't allow to see admixture, everything looks monochromatic. So this kind of is a little sign showing that you can actually track human migrations tracking H. pylori genomes in the infected individuals. So what we hypothesize is that there are parts of the genome that, because it's an admixed bug and highly recombinogenic, that may have had horizontal transfer such that they change the virulence in some people. And so there are, you can have admixed humans, you can have admixed H. pylori. And therefore, you can actually potentially examine those. And in fact, the ideal natural laboratory for that is the New World, where there are people from all over the world. Recent migration from Asia, the Amerindian population, European migration, and Africans migration. So what is this Latin American high altitude enigma? So this is a well-documented thing, which is true in the population we're talking about. In a lot of Latin America, infection rates are high, but if you look at a coastal region versus a mountainous region, the prevalence of gastric cancer is about 25 times higher. This has been well replicated in multiple populations. And so the question is, so the thought from the epidemiological point of view is there's something about the altitude in those mountains that changes the pathogenicity and the progression to cancer in those populations. I will show you that's not true. So I worked with a group who'd been working in Colombia, South America, for a couple of decades, following a very large cohort of people who presented with gastric symptoms. They biopsied them. They also happened to draw blood from the same people they biopsied, and that's a really key feature to the study. It was a natural laboratory. So there are two areas. The lower area here is, on the left is Tumaco on the coast, and on the right is Tuqueres in the highlands, in the Andean highlands. And although I don't want to generalize too much, although you'll see the genetic evidence, the people on the left look more African, and the people on the right look more Amerindian. And in fact, that coincides with their genetic ancestry. So we have this population. So what did we do? We had, and this is despite the fact that more data is usually better. If it's good data, it doesn't have to be so large. We had 233 paired samples. So we have the genomes of the bug and the genomes of the people. We did the immunochip on all of the humans, and we sequenced those 7 housekeeping genes in the H. pylori. And we compared those to the databases in terms of geographical countenance. So if you look at the human ancestry, in fact, it's about 60% African in the coast, and a little over 60% Amerindian on the mountains, with low levels of African human ancestry in the mountains and moderate levels of both European and Amerindian ancestry in the coast. If we do the average of the H. pylori on the coast is predominantly, over 50% on average, African, Africa 1, which is West African predominantly. And on the right, it's the H. pylori is predominantly European. And we could talk about what that means in terms of there was actually once an Amerindian strain that has been effectively replaced by the European. But then there are moderate levels of African ancestry in the H. pylori. And we can look at this on the top. These are the H. pylori ancestries from our sample, 233 samples. On the coast on the left, you see there's a lot of blue, which is Africa 1, and in the mountains, you see a lot of green, which is European. And this is what we did, was we took and we lined up, each line now is a pair. The top is the human ancestry and the bottom is the H. pylori, and we ordered it from left to right with respect to human with Amerindian. And the mountain is more Amerindian, as we showed with the means, and you can see there's a lot of variation around the H. pylori. And here we can look and see that there is in fact a correlation. As the human ancestry increases, which is the x-axis, the African ancestry decreases, and the European ancestry increases. So there is some correlation. There's also some relationship between that and diseases I'll show you in a second. But what we did is, my colleagues who were been working on this project for years, I will tell you that they came to me and said, we have these samples, they're paired, what should we do? We have money. Those are the kinds of problems I like. And so we did the genetic study this way. They took everyone who was over 40 and they looked at that cascade that I showed you, which has now been worked out to be quantified on a scale. It's actually from 1 to 6, 1 being no symptoms, 6 being gastric cancer. They had multiple pathologists look at it and come up with a score for everyone who was over 40. And we then did regression analysis. We also did ordinal regression analysis. And the outcome was the histopathology score, this score from 1 to 6. In our case, it was 2 to 5. And then we looked at multiple explanatory variables in a univariate analysis. First, we looked at the mountains, location. Higher in the mountains, it's exactly what we expected. That's what everyone has seen. CAG-A, this kind of virulence factor, presence of it associated with worse disease. But then we looked at all 3 human ancestries, and all 3 of them associated. But the thing that's interesting is the Amerindian associated with the beta was positive, which means the scores got higher or worse disease. The African, the scores got lower, meaning less severe disease. And they were both highly significant. But when we actually only looked at the coastal population, we saw exactly the Amerindian ancestry associated with significantly worse disease as well. Now the p-value is not as small, but it's also half the sample size. So that's telling us that there is something to do with the human ancestry. Now what I'll tell you is when we looked at the H. pylori ancestry, we saw no association. But that wasn't too surprising, because if you look at the bottom of this slide, the African H. pylori, if you looked at that ancestry, only looking at the mountain human populations made things worse, but the European ancestry and the H. pylori made things better. Duh. That was the kind of light bulb, although it was derived a slightly different way. And we said, why don't we do a multivariate analysis and see what's really driving this? So we put in multiple covariates. We looked at CAG-A, we looked at location, we looked at Amerindian human ancestry and African, and we looked at an interaction term between the human ancestry and the H. pylori ancestry. And I may run over a minute or two. And when we did that, the interaction term was significant. But the beta value was 5 times the beta value for the CAG-A, which was the bug pathogenic gene that had already been known. But the other thing is, in that adjusted model, with the interaction term, Amerindian ancestry went from a p-value of 10 to the minus 3 to not even close to significant. Location, which was statistically significant in the original, disappeared. So what was driving the pathogenicity was not the ancestry or the genetics of the host or the genetics of the bug, it was the genetics of them together. And you can actually see that again in the mountains. The African ancestry made things, the H. pylori made things worse, the European better. So the question is, what's causing this African enigma, or this Latin America high altitude? And we concluded that it's not the presence of the bug, it's who you are and what the bug is that is driving the severity of disease. So in fact, we concluded that the interactions between the human and the H. pylori genetic variation confound the effects of each other on histopathology scores, and the evidence of functional relationships, which is evidence of functional relationships or past coevolution, where they have been commensal, they have co-evolved to be benign. You admix both the human and the bug, and you end up with an increased risk of severity. I may need another minute or two as well. So what we concluded in this is that host and pathogen genomic variation are more predictive of histopathology score when analyzed together than when either one is alone. This model is a clearly interaction across species. So, does this have clinical relevance? And this will be the last slide. And the answer is maybe. Because I started out by saying you don't want to eradicate H. pylori in everyone. Well, if we know something about the genetics of the bug that a person has and their own genetics, we can actually predict when they are discordant, you want to eradicate, but if they're concordant, you don't bother touching it. And this is just by basically saying if you took the 95th percentile of Amerindian ancestry, human ancestry, if you have high African ancestry in the bug, your histopathology score is almost 5. If you have low African ancestry, it significantly drops to 3. And remember, everyone who was involved in the study was presented with clinical symptoms, so they all have reasonable. You take the flip, and you actually, with the Amerindian, with the European, you lower it. So the point is, we have a mechanism, although this is not the most precise mechanism, of determining, yes, there are
Dear
some
doctor,
people...
your time is up.
Okay. There are some people, in fact, who are trying to eradicate. Can you at least acknowledge the people who did the work? So what happens is, we now have a clue as to what is driving the pathogenicity of this particular bug. And in fact, it's not just Helicobacter pylori that has evidence for coevolution. 2 minutes. Okay. There's evidence of coevolution in human papillomavirus, Mycobacterium tuberculosis. So the point is, if you look at the microbiome, yes, you may have the right species, you may have the wrong species. You probably have, what determines pathogenicity is probably more refined than the presence or absence of a species or a genus, which is how we usually assign the microbiome. But what particular strain of that bug do you have? All right, so the bulk of, the lion's share of the work on this project was done by my graduate student, Gary Kodeman. These are all the people who have been working on this project for over 20 years. They've been following up this cohort now. It's on its 5th cycle as a program project. And what I want to say is that we are now doing a replication of this in Honduras and the Caribbean coast, the highlands of Honduras, and the Pacific coast. But instead of doing the way we did before, we're doing full genome sequencing of the H. pylori and GWAS. So we'll be able to not only look at the ancestries, but we'll be able to start mapping the genes that interact between the species. And that's where I'll end. Thank you very much for your time.
Thank you so much, Dr. Williams, for that really interesting and excellent talk. Does anybody have questions for Dr. Williams? There's a couple of questions.
Thank you very much, Professor, for your interesting talk. I have a question about the coca use of the high altitude and its relationship with the H. pylori. In high altitude in South America, I think coca use is used for medicinal and cooking and several things. So, yeah.
Can you
I
repeat your question?
Yeah, I was asking about the coca use in the high altitude population and its relationship with the H. pylori evolution.
The relationship of the human population with the H. pylori?
Yeah, the human population in high altitudes in South America usually use more coca and coca plant, the plant which contains the cocaine. It's normally used in cooking and different medicinal ways. Well, would this be a relationship? Actually, it's sometimes associated with some cancer, high risk here.
Theoretically, it could be, and as I pointed out, there are environmental parameters which affect the susceptibility to gastric cancer. However, the point that I will make is that, even though the environments change between the coast and the mountains,
Yeah.
that interaction made that difference between the host and the mountains non-significant. So basically, even if there were differences that affected around the edges, it looks like the genetic interaction is what's driving it.
Okay, thank you.
Ambrose had a question in the back.
Hi, thank you for your interesting talk. I was wondering about the relationship between H. pylori and liver cancer. some people said there is an association with liver cancer for H. pylori. So, what is your comment on that? Is it real or not?
I actually don't know about that, so I won't even speculate. And I know that the liver cancer is worldwide generally relatively rare as compared to gastric cancer, but I know it's not that rare here based on some of the talks I've heard, but I do not know the answer to that question.
Thank you.
Thanks, Scott, for a very fantastic program and presentation. I think there seems to be a parallelism between what you just presented, Helicobacter pylori, and the co-evolution of TB and human genome. And to comment on that and how can you draw information from one to feed the other?
So you were talking about tuberculosis and the interaction. I think there's actually the same kind. There is some evidence. Sebastian Gagnot's work suggesting that, and Erwin Shor's suggesting that there is in fact coevolution between mycobacterium and the human host. Now there are a couple of things that one needs to consider. Mycobacterium is not as recombinogenic, but because it's airborne, it moves quite easily across populations. Now, that said, I've been trying to get funding to study it, and we haven't succeeded yet, but I actually think that that is a really ripe area globally.
Thank you. Seems like there's lots of questions. Maybe one last question? Couple questions.
Thank you. Thanks you very much for your interesting presentation, but I'd like to ask something. Nowadays, we hear a lot of researchers that highlight the relationship between whatever the disease and the ancestral origin. But what is the next step after that? Can we highlight specific polymorphism or genetic factors that can affect the interaction between the human and the parasite?
Yeah, the answer is yes. We couldn't do it in this study because we only had the sequencing of 7 housekeeping genes. But now that we are funded to do the full genome of the H. pylori and Ben's GWAS, and we're doing it. So the goal is to go gene by gene across the 2 species, step by step, so we can identify the genes that are driving this relationship. It's not the ancestry per se. It's got to be some genes that are differentiated among those populations, and we really want to map those.
I hope so, because Doctor, we can find the relationship between two totally different things, and we can prove that there is a relationship, but we cannot be confirming there is a causative effect of that relationship to affect that.
Well, in this case, I mean, I think there's evidence for a functional relationship between those genomes. We just don't know exactly what it is yet. The ancestry is a surrogate. But the point is to get to the variants in those 2 genomes that are driving us, and that's where we're going.
We have so. Thank you.
Okay, I think we have to close. One more quick question.
Thanks, thanks, Scott, for illustrating 2 of the things I always think, which is we need to do a much better job of looking at host and pathogen genetics together rather than separately, which is what we usually do. And then a well-designed study can show a lot, even though it's small. But my question is, what is sort of the evolutionary or co-evolutionary time scale that you think this type of study will be effective for?
Well, of course, the 2 genomes evolve at very different rates. But based on some of the things that I know about how quickly humans respond to infectious disease, I mean, the G6PD variants, 5,000 to 10,000 years. We're not talking enormous time periods. So we're probably in the zone of that similar range. And of course, given an infinite amount of time, we would probably see things evolve back to a benign state globally.
Thank you so much, Dr. Williams. I'd like to thank both the speakers for this session, Dr. Barbouche and Dr. Williams, for an excellent session. And I'm sure you can find them during the break for more questions.